Good afternoon, everyone. I'm Sean O'Neill. I am the Compliance and Privacy Officer for Vault Workforce Screening. Vault is a trusted workforce screening partner supporting clients ranging from government agencies to Fortune 500 companies in the design and management of their screening programs for the last 34 years. I want to welcome everyone to our webinar today, which is called A Closer Look into Laboratory Testing, a Day in the Life of a Urine Sample, which is going to be hosted by Dr. David Kuntz. Dr. Kuntz is a PhD. He's the Executive Director for Analytical Toxicology at Clinical Reference Laboratory in Kansas. A Board Certified Toxicologist, he specializes in drug testing using advanced methods and has over 35 years of experience in the field. Dr. Kuntz has developed tests for synthetic substances and provides analytical support for federal studies on marijuana and CBD. Following the presentation, we will have time for Q&A. On behalf of everyone at Vault, we want to thank you for choosing to spend your time with us this afternoon. And with that, we're going to turn it over to our featured presenter, Dr. Kuntz, and we will get started. All right, Dr. Kuntz, are you seeing my screen?
I am not. All I see is me right now. I have slides printed. I will go from paper. If the audience can see the slides, that's great. Let me begin. I'm going to give you a 35,000 foot level review of the process. And we're going to go through this testing process. And now we'll show you just quickly the front of CRL. We're a multi-building structure, and that's just the corporate headquarters. CRL is a privately held company, and that's worked out very well for me professionally. We're one of the largest privately held clinical testing laboratories in the United States, and one of the largest workplace drug testing laboratories as well. We are a HHS DOT certified lab or CAP accredited states wherever we need to do. And with all of that, the laboratory is always extensively reviewed by external people. For instance, like the NLCP, National Laboratory Certification Program, we're up to 7 people, 4 days at a time, 4 times a year. That is a lot of people looking at us just from the federal program. We have our College of American Pathologists annual inspection, and then we have NRC and on. The drug testing processes are all very well reviewed for decades. And people out there should have great confidence in the laboratories to get the results accurately analyzed and reported. Now, going on to slide 4, the sample receipt process, that is a big job every day. Moving on to slide number 5, I've created these slides with some descriptions on the side, along with some pictures for each one of these. So we get into sample delivery. Our primary sample deliver, like most laboratories, is Federal Express. And since we are a large laboratory, we get these bags of samples, if you will, in the FedEx, and there'll be 3,000–4,000 bags a day coming into the laboratory. And a bag could have 1 sample or it could have a dozen samples. You just never know, depending on the day of the week and the clinic and which company you're working with. But the vast majority of the time, our samples do come in between 7 and 7:30. We also have 60, 70 different airline markets that in different cities that send samples to us overnight with flights like United, Delta, Southwest, and so forth. They get here early in the morning, typically by midnight thereabout. Moving on to the next slide, number 6. Now, the samples themselves, or the bags, are all scanned and they're placed in those bigger plastic bags. They're like an 80-gallon bag. They're like a jumbo trash bag. That's clear. And there's tags on those. And underneath, in that one big bag, there may be 10 to 30 different packages, and we scan that so we know what's in there. But we also receive boxes in slide number 6 that we also scan. that would have an individual box, for instance, or an individual bag to provide that tracking. We do all of that every day before we even get started. Slide number 7. There's a lot of teamwork that goes in that, and it doesn't do it justice with what you see there. We have actually a automatic table conveyor belt, if you will, that moves samples from one end of the room to the other so that we can begin the opening. It's a big process. It takes 15 people from 7 o'clock in the morning till 10 o'clock in the morning just to get rid of this exterior packaging that FedEx is shipping in. A lot of people are involved. As you can imagine, it all goes fairly quickly. There's a lot of pressure to get them out so that we can sort them, and then we'll break them out into varying testing groups. For instance, federal DOT goes one way, the nuclear regulatory goes another. We have some railroad types of things. Oral fluid goes another direction. So it all gets sorted correctly so we can begin that process. But with all the packaging, we also have a great deal of concern about ever missing something. And that can be found on slide number 8. We have an individual looking at every package that we open. It's re-inspected before it goes out into the trash. We want to make sure that every sample that's been sent to us, whether it's an oral fluid, whether it's a blood, whether it's a urine, that we extract them. And with the packaging, it sometimes is a little bit difficult to get them all out because you got that adhesive from the seal from the bags. They all like to stick inside into that plastic. We do spend the next 2 hours, I think there's 5 people researching every one of those bags, again, looking for some lost paperwork, a sample that got stuck in there because of adhesives. Moving on to slide number 9. This gets into a little more fun. We'll talk a little bit about accessioning, receipt, and pouring of the aliquot. Now, aliquot's one of those scientific terms that we use in the drug testing laboratory, which means it's nothing more than a portion of the original. So, that original bottle that we always call bottle A, bottle B is reserved for the donor. And I'm sure you're all aware of that for DOT or even other testing, but it's for non-regulated, all you have to have is a bottle A as a minimum, and there's requirements there. Slide number 10. Now, when we get into looking at all of this, we have to make sure it meets the chain of custody requirements, because we have to make sure that every result that we turn out is forensically and scientifically supportable. We're going to be looking at a series of criteria. This is just a few, just to make you aware what every time they open up those little clear bags that have the bottle and the chain of custody, what they're looking at. And that would be such as, does the form in the bottle barcode match? Obviously, that's the only way that we, the laboratory, knows that the bottle and the bottle and the paperwork match, because very often we don't get names, or if we get initials on the label, it's almost impossible to match up names, and even under the federal program, it's prohibited. Now, is there a sufficient volume of urine to test? Are the seals intact? Does the collector sign the paperwork? Is the color in A and B the same? Now, we can have all kinds of different colors of urine. It can be clear to yellow to orange to blue. And as long as they all match, bottle A and bottle B, we're instructed to test. That's a federal requirement. But if bottle A is yellow and bottle B is green, that's a problem, and there's another side route for that. Now, because we open all of these to get to the urine, we can smell them. Sometimes you can smell bleach if they're intentionally adulterated, but that's not very common. Opening, you really can't tell whether it's a synthetic urine or whether it's real urine, but we always know that the real urine always smells worse because synthetic urine is usually water plus chemicals added to mimic a urine sample. And then, of course, if it's a federal sample, is it the right form? If any of those things, there's the memorandum for record that we use, or memorandum of correction, MOC or MFR. And of course, that always ends up a delay in testing. That typically always goes back to the collector, and they work that into their portion of the day, getting that resolved and sent back to us. We move on to the next slide, number 11. That's just a quick image of an individual scanning the form, and so that we can obtain the beginning of the demographic information. We're going to have the upper right-hand corner is usually the custody control form number, this big long barcode. We are going to be applying, as you can see on the desk, there's another strip of numbers, a secondary number for ourselves for at the lab to allow us to track these effectively. So we're going to go through that, and that's part of the slide before is what they're quickly scanning for completion. Moving on to number 12, the next one, that is just a simple image of checking the barcode on the bottle versus the CCF. Now, if they do not match, if bottle A, bottle B, and the chain of custody, if we scan and they do not match, there is no moving forward. That sometimes can be a problem if collections get out of hand, such as you may have more than one individual slowly providing a sample due to shy bladder, or if the seals tear and you try to recover it by putting seals on over top of seals. And I understand all those things, but if they don't match and we do not find the justification for why they do not match, obviously that would be another rejection. Now, the next slide, 13, this is an example of pouring the aliquot, and it's a little bit of a strange word for people. It's unique to the testing laboratory that was created in the late 1980s with the program. And what the individual is doing is the bottles are typically all the same. They have the flip top lid, and they're going to break the seal with their finger. They're basically going to pop it. And that's, you can see the lid is still adhered to it. And that tube that she is pouring into, I assume it's one of the female collectors. I said she, we both have male and female. They're going to pour about a ½ an inch of urine into that tube, which is about 2 milliliters. A bottle typically will have up to 45 mils. There's more than enough urine to do all the testing that could ever be envisioned for this testing. There is a circle, a yellow circle up at the top of the page, and this is what we will refer to as creating the batch. And a batch is just a collection of samples that are included one after another, because under chain of custody, everything needs to be documented. And that's what we're doing here. Instead of doing 20,000 signatures a day, we're trying to cut that down to maybe only 500 for a batch. And so a batch will contain 35 or 40 samples that we would use for testing. It is a simplistic, simplifies it. It's endorsed by the federal program forever. That's how the program started, and that's how we work. Now, going on to slide 14, we see an individual with a chain of custody, and then at the end of screening, all the bottles get stored. as you well know, we are storing all of our screen negative samples for a minimum of 5 days. And if it screens positive, it stores up for a minimum of 1 year. Now, every now and then we do get requests for extended storage, whether that's going to be 12 months or whether it's going to be indefinite. I know I have samples in the freezer because of litigation of some nature, maybe a post-accident or just a drug challenge, that have been back there for 20 years. They stay. We will retain it if you ever need to have it retested, whether it needs to be inspected for initials and dates and all of those wonderful things that can occur. And moving on to slide number 15. Excuse me with the cough. We're going to get into the screening lab and moving on to 16. That's an image of a part of the screening laboratory. The screening laboratory is our fast way to obtain test results for individuals that are completely negative. If you have a 10 drug panel, 5 drug panel, all of them are negative, they're going to run through this instrument very quickly, and we're going to be able to report that in a matter of a couple of hours. If it's positive, it goes through an alternate track, as you probably all are well aware, with confirmation. But that's just a quick example of a picture of the laboratory. Each one of these instruments is very expensive. They do tremendous amounts of work, and it's the same basic instrument with different reagents, whether you went to the clinic and had your blood drawn and you're going to have your cholesterol tested or any of the other things associated with your blood chemistry. They all go on the same platform, but in a clinical lab, they'll test for your cholesterol, they'll test for your potassium, and I'm more worried about your marijuana and your cocaine and your methamphetamine. That's what that's all about. Moving on, this is a little closer look on slide 17. The batch requirements, we follow the same rules, whether it's a DOT sample or whether it is a non-regulated sample. The instruments are all calibrated. There's a minimum of 10% quality control. We do the specimen validity test and we do the data review. And those little tubes in the batch go through that analyzer. And looking at slide 18, which is your next one, gives you a kind of an inside view of this instrument. And under that circle, there is a probe. And that goes and it will sample reagents. There's another probe that for a separate reagent. There's probes in the back to sample the sample. There's like a little spaghetti tubing in the upper right-hand corner that is basically a little janitor crew that will clean and rejuvenate each one of those cuvettes that's performing the testing. It is a fully automatic testing machine instrument that does a tremendous job. The advantage of all of this is that we can test quickly. And I wrote in there we could do 3,500 samples an hour, not tests, individual people. We could do 3,500 people per hour going through the instruments that we have. For those that are concerned about testing volume for laboratories, the instrumentation for screening is tremendous. It can do a lot of very good things. Moving to slide 19. Now, those that have worked extensively with CRL over the years know that we have a lot of additional tests that are not your traditional chemistry tests, such as marijuana, cocaine, methamphetamine, opiates. We do a lot of other things, such as K2s, bath salts, antihistamine, alternate narcotics, you name it. We have a process to assist you in your testing needs. We have another instrument that will sort that for us and put them in order so that we can do our K2s. synthetic cannabinoids. Those are primarily still used for oil and gas, but the other things like narcotics and antihistamines are used by people in professional monitoring programs and so forth. We can cover both spectra, whether it's regulated, non-regulated, specialty testing, and so forth. Slide number 20 is the breakout for specimen validity testing. And then slide number 21, I'll comment a little bit about pH. pH is the measurement of acidity or basis of whether the sample passes or not. Every summer, we have a
We are working to get Dr. Kuntz back online here. We ask the audience for your patience and please stand by.
not fresh and cold, the pH goes high, and we term that invalid. And we've traditionally, it's been a pH of 9, which is slightly basic, and we introduced a different scale here at CRL. We changed it to 9.5 so that we can get more of these donors hired, because the samples are negative. There's no drugs, it's just the pH. And through the 1990s, the federals established that limit, and we've been stuck. Slide number 22 deals with specific gravity, and that's just a little handheld device to determine whether you're dilute or not. Those are little handheld tests that we have to do for every time the urine pH is at normal, whether the specific gravity or the urine samples appears to be dilute. Those are all additional manual tests that we do. Now, moving on to slide 23, it just breaks up the section. We'll talk a little bit about mass spectrometry, which is very close and near to my heart. And looking at slide 24, now, nearly 10 years ago, we introduced liquid chromatography mass spectrometry to the laboratory. And this is a state-of-the-art system. LCMS, GCMS all provide the same answers. It all provides you the correct result. It's just a matter of what kind of technique you prefer to use and how much effort will go into a sample preparation. At CRL, we chose to go with the more automated way. And on slide number 24, we have a row of tubes. As you can see, there's 3 rows. When we have a positive sample that's screen positive, we go back to that original container, and you remember where we had the individual pouring urine into a tube. We're going to create a second tube that we're going to use for confirmation testing. It's two takes of the same bottle, one for screening and one for confirmation. But we chose to use robotics as our way to get samples ready. And moving on to slide number 25, I have these big black probes, and they're pipette tips. And what they're doing is sampling the urine from the tube, and we're going to move it into a separate location, into a plate that we can do some confirmation testing. This is all robotic, manually handled, and we do a lot of confirmations here every year. We do about 1 million a year without fault, and having tired people making a mistake. That's one of the reasons why we did this approach, is that these robotic liquid handlers are awesome in their ability to transfer liquids. There's barcode readers on things. Everything is documented. I'm not relying on a newly trained person to get the right result, or someone that stayed up too late with a child who was sick last night, whatever it would be. It's always the same. Moving on to slide 26, that liquid that we pulled out of the tubes move into what we call a deep well block. And that's ultimately, we're going to put the other remaining reagents for testing for confirmation. And that process works extremely well. And we've had 10 years of evaluation by the federal program, and they all know it's effective. It works well. Again, everything is documented. If it can't read a barcode, for instance, or something touches it, it automatically stops. It is the safest, most secure way for me to do your testing that's possible. Now, moving on to 27, it's just a quick picture of the lab. Everything as I mentioned, is LC mass spectrometry, tandem mass spectrometry. We have 40 of these instruments. I have this million samples I got to get done every year. And we do work 7 days a week, 24 hours a day. We do take a couple days off. It's like Christmas Day and those types of things, but almost every holiday except those there's people in the lab working to get that work out. On slide 28, there's another, that's the inside of the mass spectrum of the liquid chromatograph. Inside the circle, there's the analytical columns, and that's where the separations occur. We can have hours worth of lecture on that as well. But again, this is just a kind of a top level. Now, the important part for this, what I wanted to portray to you, is that each one of those silver tubes, if you will, is an analytical column, which means that this instrument is set up to run 5 different assays. The GCMSs could only do one. We gained automation, we gained versatility by doing it in this way. And this is the standard way that the pharmaceutical industry is working. The clinical labs are doing it this way. We're not a trendsetter anymore. We're part of that. We are part of the group. And page 29, or slide 29, is just the mass spectrometer. So unimpressive. It looks like a giant blue and tan box, but that's $400,000 right there. It will look at all the chemicals in your body. I can tell you everything that's in you, whether it is a drug of abuse, whether it's a clinical sample. I can look at your blood chemistry. It's a jack of all trades. I train it, I teach it, I certify it to do certain things. If I want to do environmental chemicals, we can do that. If we want to do pharmaceuticals, we can do that. But we are just doing drugs of abuse. And as we get close to the end here, we get into certification results on slide 30. One of the things I wanted to relate to the group is that there is dual certification at each step. When we screen samples, whether it's negative or presumptive positive, it gets reviewed twice by people. If it comes into the confirmation lab, it gets reviewed twice by people. And then if we look at slide 32, what they're looking at, this is just a little piece of a image of a marijuana chromatogram. So they are looking at that, and each one of those little colored peaks is a piece or portion of a marijuana molecule. And we look through that. We have all of our QC, all of our forensic parameters that we have to meet. But that is what the inspectors come to look at as well, to make sure that the laboratory has done everything within their power. And then as we finish up on slide 33, we have our final certification. And this is the 5th person that will be looking at everything that the others would have for positive. They're going to be taking a look at the chain of custody. We're going to take another look to make sure that it's correct. We're going to look at the confirmation data, the screening data. We're going to do a final QA of the process before it's released to the MRO. And that's probably that high level look. And 34, I just finished up with the picture of CRL, and 35 is a big old thank you for listening to me today. And I hope I helped you understand a little bit about the process, and perhaps in the near future we can repeat and we can have one volunteer to do one with oral fluids, since that's becoming the hot topic if we ever get that approved. That whole handling process is different than urine, obviously, but the testing, whether it's urine, the screening will be almost identical, different reagents, different cutoffs, different drugs, and the confirmations will be the same. It's that middle piece that we can probably have another seminar on, and we can explain how do we get that liquid out of that tube and into the testing lab. So thank you very much.
Dr. Kuntz, thank you for walking us through that. We'll give your voice a second here to rest and get you an opportunity to get a drink before we jump into Q&A. At this point, we are opening it up to questions for Dr. Kuntz about the laboratory process for urine. Dr. Kuntz, I did want to ask, going back to pH, we had a momentary audio issue where we lost you for about 15 seconds. You were talking about pH and summer. Can you just fill in the gap there? What happens or what we experience in summer in high temperatures with regard to pH of specimens?
Certainly, I'm happy to go over that. It is a major issue for pH. And just to reiterate a little bit, the urine by nature has some bacteria. We have natural products that we excrete through the kidneys. There'll be some glucose and there'll be a little bit of protein and so forth. The bacteria like it, and so they try to metabolize it and grow, to expand their colony, if you will, colony of bacteria. And when they do that, they're producing, usually as a side product, ammonia. Bacteria like heat. The warmer it gets to a certain point, 100 degrees, that's your body temperature, 98.6. The bacteria grow very well. What we discover in the summer, there's always a natural level of samples that will run higher, pH of 9. That could be due to a bladder infection, could be due to a male with prostate and the issues and you can't empty your bladder. There's all these things that can happen. But in the summer, it goes up about 4 to 5 times that rate. And so by choosing a pH of 9, and there's a great fundamental reason for that in 1989 when that was created. The pH that normally occurs, especially samples that are collected like on a Friday, don't get into the laboratory, they get to a pH of 9.01, 9.1, which is just barely over the line. And so based on following the federal regulations, we were reporting those as invalid and they would need to be recollected. And that was not due to any effort by the donor to try and subvert a drug test. It is basically temperature and time to get to the laboratory. We made some adjustments in our status for those accounts, those clients that wanted it, but it normalizes it by season. So January looks like July and August. And that's what we were attempting to do to protect jobs.
Great, thank you for covering that. Okay, we do have some questions coming in for you. So the first question is, has CRL seen lower sample acceptance rates due to higher drug positivity since the COVID pandemic? Any trends, Dr. Kuntz, you've seen since the pandemic, just with regard to drug positivity?
The positivity rate, probably the one place that, refer you to is the competitor, obviously, is the Quest Index, drug index. I think almost everywhere, I think there's slow increases or rapid increases. The marijuana's pretty much everywhere. If they're testing, I have 800 positive marijuanas every day, which sounds like a lot. And it is. It's a lot of marijuana positives. Those rates have gone up as legalization and over the last while. Methamphetamine is going up, cocaine is going up. With fentanyl, based on the data that we have, 2 years ago, we didn't see almost, or maybe even 3 years ago, we didn't see any fentanyl. Now, in our urine population, for those that test for fentanyl, it's about 1.5%. That's a little over 1% in our oral fluids. I would imagine a few of those are medical, that there are some people out there, but the vast majority of them are illicit fentanyl usage by consuming a product with fentanyl, whether that's going to be an OxyContin or an oxycodone with fentanyl, heroin with fentanyl, methamphetamine with fentanyl, cocaine with fentanyl. It seems like it's everywhere. It's the extra ingredient that you don't really need, and it is quite so scary. One thing, I did a WebEx a couple weeks ago for SAPA, and I did some background on that. I think from Customs, U.S. Customs at the border, I think it was 2016, I think they had 20 pounds of fentanyl they found that year. I think this last year is like 23,000 pounds, which is about confiscated, which is about 5 billion doses, not billion, billion doses of fentanyl. Fentanyl is the next drug of concern, but all of the drugs seem to be going up, but the hydrocodones, oxycodones seem to be fairly stable. Those have dropped, especially at DOT, because of the safety concerns with opiates.
Great, thank you. And I did see HHS is looking to discuss fentanyl at the next DTAB meeting for potential inclusion on the federal panel in the near future.
Yeah, that's true. We did some of that background work. I'm trying to remember the exact number. We did 300, 500 opiate samples that were positive. These are codeine, morphine positive samples. And we did a deeper look into those samples just to give you an idea of what's going on out there. And they didn't trigger positive for heroin or fentanyl. But when we looked at those samples and we looked below the cutoffs, I think it was like 15% of them had fentanyl. Another 20% had heroin in low doses. The numbers were big. They were alarming about the amount of narcotics that were probably being bought off the street, obviously, if they're contaminated with heroin and fentanyl. Yeah, we're still losing 70,000 people a year, I think, to fentanyl overdoses, and another 30,000 to the other narcotics. 100,000 people a year are losing their lives to narcotics, and what a national shame that is.
Yeah, most definitely. We do have another question. What are the most common over-the-counter or prescriptions that may cause false positives during the testing process? Is there a commonality with regard to false positives? As you walked us through all of the testing, the robust testing that is done, how does that play out with potential false positives?
The screening reagents, the first test, actually work quite well. Let's pick on marijuana first, okay? It's the number one drug.
Sure.
There are an over-the-counter prescription drug, I should say, Protonix, which is an acid inhibitor for people with gastric issues. That will trigger a false positive screen, but it never confirms. We all know about it. It's been known for the last 25 years. There's the one HIV treatment drug that can cause a false positive screen. But those are the two prescription drugs. You get into CBD That's always one of the questions. CBD by itself doesn't really cause a problem at all. It's when you get impure CBD or something along that line. Now, if you are Delta 8 user, which is legal in many states, even here in Kansas where I live, that will trigger a screening positive. It definitely will, because it's such a minor variation. But we separate all of these things out, and we are not reporting Delta 8 THC to our clients unless they specifically ask us for it. There's really no concern. Even we're in the cough and cold season now. We're getting the spring, it's allergy, and everybody's pseudoephedrine this and that. Or if you're overweight, you could take phentermine or Diedrix. And all of those types of things can screen positive, but they confirm negative. And that's why we do the confirmations. And we know those targeted compounds. You go to the dentist, the prilocaine, bupivacaine doesn't cause cocaine positives. We know that. So the risk is exceptionally small. That's why there's also bottle Bs if there's concerns, if a laboratory has made a mistake. But we do everything in our power never to get to that level. But there are those safety nets are built into the program, whether it's a federal or not, to help people.
Great, thank you for walking us through that. We did have a comment come through, Dr. Kuntz, that this person did not have a question, but she appreciated the training. It is helping her to better understand the steps in understanding this for her job. Definitely the content has hit its mark, so thank you.
And I appreciate the offer to be able to do this. And if you want to share the slides with people, that is certainly fine with me. There's nothing proprietary that I believe that's in there, and you can use that as warranted in whole or in part as you see fit.
Great. We appreciate that, Dr. Kuntz. And clearly from the comments we're getting like this, the content has hit the mark. So want to thank you again for joining us today and spending time with our clients. I want to thank everyone today for attending. Again, thank you, Dr. Kuntz, for jumping on with us and delivering the content that you shared with us. I know I learned a lot from it. Want to express to those who attended, if you are using Vault for your drug and alcohol testing services, we hope today's presentation leaves you with a sense of security. You made the right decision in trusting your needs or expert team and the partners we work with, Clinical Reference Laboratory. If you're considering moving your drug and alcohol testing services to Vault, we hope that you see the experts today from our partners like CRL understand the nuances of testing and will help you keep compliant with federal, state, and local laws. If you have any questions that come up following today's presentation, we're still available to help and would love to hear from you. Please drop us an email at compliance@vaulthealth.com, and we'll make sure it gets routed to the appropriate person. Again, a recording of today's session will be circulated to you via email very shortly. Again, Dr. Kuntz, thank you for joining us today. Really great content today, and I really think it helped educate those attending just what it is to be a day in the life of a urine specimen going through the lab. So again, thank you very much for spending time with us today.
It was an honor for me to be able to join you. So thank you.
Great, thank you. And I will take you up on your offer to do another one on oral fluid as we get closer to, as we get DOT closer to making that available. We are ready and we will gladly take you up on that invite. I'm going to hold you to that one.
I'm sure that it was, many people would like to see something like that. We can just talk more about oral fluid, how we prepare them, and those aspects, because it is substantial. But the testing process, the front and back end's the same. But it's the middle that's the important part. How do you get it in a lab? How do we get it out of that little tube? And how do you make it all work, right?
Yeah, great. I look forward to that. All right, Dr. Kuntz, again, thank you for joining us. Thank you to our attendees for spending time with us. On behalf of everyone at Vault, I want to thank you very much for choosing to spend your time with us today. And we hope you have a great rest of your week. Goodbye, everyone.